A lot happens to fish before it ends up on your plate, and it's shadier than you think. Few outside the seafood industry know about the worldwide epidemic of fish and seafood mislabeling and substitution. This little secret generates hundreds of millions of dollars annually in the U.S. alone. It's a low-risk, high-return endeavor, and as always, it's the duped consumer who pays for it. At the root of the problem is the simple fact that once a fish gets gutted and processed into smaller pieces, it looks like most any other fish, even to the expert eye. Therefore, it is easy to substitute any fish with a less expensive look-alike. Despite its pervasiveness, the fish and seafood mislabeling problem is rarely brought to the attention of consumers. In the past year, both the New York Times and the Boston Globe had splashy exposés, but these were the exception rather than the rule. The bottom line is that most of the public is still unaware of this lucrative scheme. Fish and seafood mislabeling and substitution can occur at many levels, because the fish changes hands numerous times before it gets to the consumer. This type of product tampering is plain and simple fraud. Those who turn a blind eye in collusion are equally responsible, because we now have tools for accurately testing the authenticity of all processed fish and seafood. Today, DNA tests can determine the species of fish and seafood sold on the world markets. Although the mislabeling and substitution of fish and seafood is a global problem, we are curious about its prevalence on a local scale. We decided to test the authenticity of fish and seafood sold at several popular and critically acclaimed sushi restaurants in Chicago. A DNA test was used to check the authenticity of fish and seafood purchased from 4 different restaurants. The test is simple. It involves decoding one or more genes that are unique to fish and seafood. Genes are sections of DNA that provide instructions for making proteins. The first step in the test is to purify the DNA from a small piece of tissue. From this purified material, more DNA of each test gene is made by PCR amplification. This is necessary because gene decoding requires more DNA than tissues usually yield. The amplified DNA is then decoded by DNA sequencing. This process determines the order or sequence of the 4 bases that make up DNA. Bases can be pictured as beads on a string. Upon decoding of the gene, its code, or DNA sequence as it's known, is compared to fish and seafood sequences cataloged in databases with more than 250,000 entries. From this analysis, one can easily determine the authenticity of all commercially sold fish and seafood. The DNA test takes a couple of days to perform, but it is unsurpassed in its accuracy, because DNA is the most sophisticated fingerprint nature has devised. Our survey found evidence of significant fish and seafood mislabeling in the Chicago restaurants tested. Across all of these, 1 in every 4 fish, or 25% of those surveyed, was mislabeled. Only 1 of these restaurants passed the test, and all the fish they sold was authentic. The survey showed tilapia substituting for sea bass, a freshwater fish used instead of its saltwater counterpart, and Alaskan pollock masquerading as Alaskan crab, to mention a few cases. Consumers are misled in other ways as well. We found menus that listed an item with a Japanese and an English name, but the names actually refer to 2 related, albeit different fish. Some restaurants claim to sell Norwegian salmon and Hawaiian tuna at premium prices, but there is no easy way to verify these claims even with current DNA tests. Adding the number of times the consumer was misled with the number of times fish and seafood was mislabeled, 4 out of every 10, or 40% of those surveyed, was misrepresented. The high rate of mislabeling found in these Chicago restaurants is alarming. However, it's very consistent with the rates reported by Oceana, a nonprofit seafood organization. Their survey was far more extensive and detailed than ours. They found that fish and seafood mislabeling rates between 25 and 70% across the worldwide industry. Fish and seafood mislabeling is a local problem in Chicago, especially since it was discovered in the more acclaimed sushi restaurants. The results of our survey also ask whether this problem is more widespread in light of the small sample size. What if other less acclaimed restaurants are also surveyed? Restaurants and the entire seafood industry can no longer turn a blind eye to the mislabeling and substitution problem. It is in their best economic interest to test the fish they buy, since they may overpay for products that are not authentic. Testing also helps safeguard the public's health by preventing the sale of fish that contain dangerous toxins. Cheap oil fish are widely substituted for many types of whitefish, but oil fish contain toxins that cause indigestion and diarrhea for several days in 1 out of every 2 people who eat them. More rare but still known to occur is the sale of fish with lethal neurotoxins such as the pufferfish or fugu, whose poison kills within a few hours by asphyxiating its victim. Our natural resources also benefit from DNA testing of fish and seafood. Overfishing is endangering and threatening many fish species, especially popular ones, such as the bluefin tuna and the Chilean sea bass, to name a few. Protection of these natural resources is just sensible. At the end of the day, DNA testing of fish and seafood is smart from an economic, health, and environmental perspective. So this tale is just a warning that the scallops you just ordered are a little tougher than expected, but not because they were overcooked. You were probably served shark instead. So, buyer beware.